Good day to everyone. So for this video lecture, I'm going to talk about or discuss about the next chapter on gene functions, proteins, and enzymes. So I will be talking about the genetic control of proteins, protein synthesis, and the genetic code. So with that, um, let's talk about first what is protein so that we can understand uh, the other topics that we will discuss. So when we say proteins, these are large complex molecules that play many critical roles in the body. So they do most of the work in cells and are required for the structure, function, and regulation of the body's tissues and organs. So um, parang yung protein talaga meron siyang part or importante talaga ito sa lahat na ng ginagawa dito sa body, yung mga metabolism na ginagawa ng body sa proper functioning na kailangan ng mga organs, ng mga cells, lahat na ng mga tissues, they need itong protein na ito. So, it is used for structure, function, and regulation of the body's tissue and organ. Kaya, if you can, yung isang kailangan na nutrient ng isang living organism, particularly tayong mga human beings as well as mga animals, kailangan natin itong protein. Kasi kung um, um, wala tayong protein or maliit lang yung level ng protein sa body natin, hindi tayo makakapag-function ng maayos. So, kung walang protein, this can lead to death also. So, ganun ka-importante yung protein. So, proteins are made up of hundreds or thousands of smaller units called amino acids, which are attached to one another in long chains. So, there are 20 different types of amino acids that can be combined to make a protein. So, yun yung laging tatandaan. So, please remember it. Yung mga protein natin, it is made up of these amino acids. So, ito amino acids, in a sense, it is the building blocks for protein. So, please remember na yung parang protein, itong protein na to, imaginein mo na lang na itong mga protein na to, lahat ng ito are parang um, na mga amino acids na bind together into a long structure called itong protein. So yun, nagbabind yung mga amino acids into long chains para mabuo itong protein. So, yung protein natin, ito yung functions nila. Pwedeng antibody, messenger, structural component, at saka transport or storage. So, yung antibody, it binds to specific foreign particles such as viruses and bacteria to help protect the body. Yung mga protein na messengers naman, such as yung mga hormones, they will transmit signals to coordinate biological processes between different cells and tissues and organs. Tapos, yung mga structural component naman, ito yung mga protein that provides structure at saka support for the cells. And on a larger scale, they also allow the body to move. So, yun yung mga structural protein natin. Then, yung mga transport and storage, these proteins bind and carry atoms and small molecules within cells and throughout the body. So, yun yung mga functions ng isang protein. But, sa ating subject, which is Agri 21 sa genetics, yung pinaka-importante dito is yung enzyme. So, itong enzyme na to, it can carry out almost all of the thousands of chemical reaction that takes place in a cell. So, they also assist with the formation of new molecules by reading the genetic information stored in DNA. So, yun. So, when we say yung function ng protein, when it comes to genetics for the gene, may enzyme that will assist in the formation of new molecules. Paano niya ginagawa ito? Binabasa niya yung DNA. Yung part ng DNA which is the gene, binabasa niya yung code doon, yung genetic information doon, in which itong enzyme na to, it will help create yung mga proteins natin, yung mga iba't ibang proteins natin, mga messenger protein, mga antibodies na protein. So yun, mga structural protein. So yun yung ginagampanan nitong enzyme. So ganun ka-importante yung ating mga protein. So, ano nga ba yung enzyme na tinatawag? So, the purpose of an enzyme in a cell is to allow the cell to carry out chemical reactions very quickly. So, these reactions allow the cell to build things or take things apart as needed. So, an example of an enzyme, itong amylase. So, most of the enzymes, they are parang named. Yung end ng pangalan or name ng enzyme, it usually ends with ASE, ASE. So, to quickly differentiate an enzyme from other substances, mga other hormones, ganun, usually ACE, mayroong sa end ng pangalan ng isang enzyme, merong ACE. So, amylase, ganun. So, enzymes are made from amino acids and they are proteins. When an enzyme is formed, it is made by stringing together between 100 to 1000 amino acids in a very specific and unique order. The chain of amino acids then folds into a unique shape. So, meaning not all of the enzymes are structured the same. So, it can be yung enzyme can have mga 100 amino acid, then another enzyme will have 1000 amino acid, and then they are folded. They are unique with each other since yung structure nila iba-iba. So, yun yung enzyme natin. So, yung shape ng enzyme allows enzymes to carry out specific chemical reactions. So, an enzyme acts as a very efficient catalyst for specific chemical reaction, and that enzyme speed the reactions tremendously. So, yun yung importance ng enzyme, which is doon sa i-discuss ko, merong enzyme talaga na importante in making up or producing yung mga proteins natin. So, another definition ng enzyme is that it is a molecule that speed up a reaction. So, in relation to yung mga sa genes, sa DNA, in DNA reproduction, enzymes not only speed up the reaction, they are necessary for DNA reproduction. So, yun. Yung enzyme natin, meron talaga important role in DNA reproduction at sa DNA reproduction, sa proteins, sa DNA replication, mga ganun. Kailangan talaga nitong enzyme. So with that, let's talk naman about yung protein synthesis. So, we know now that the DNA carries the traits like the hair color, eye color, comb type, presence of seeds, etc. So, sa DNA, meron yan yung mga genes na naipapasa doon sa mga from parents to offspring. And with that, doon sa gene, nandun yung parang code o na nagsasabi na itong anak mo, meron niyang black na hair color, meron niyang black na eye color, mga ganon. Yung chicken, yung offspring, merong rose comb type na chicken, so mga ganon. So, yun yung DNA. A part of it, yung gene na yun na nandun sa DNA, it carries yung trait, yung phenotype ng isang individual. Now, ganito yan. So, yung gene na nandun sa DNA, it will make up itong mga proteins na to. So, parang you could say na yung gene gagawa ng isang protein wherein itong protein, it will dictate yung production ng, like for example, itong pigment para ito namang pigment, it will parang influence or dictate yung eye color. So it is an example of yung parang overview ng protein synthesis. So inulit ko, yung gene na nandun sa DNA, it will code for a specific protein. Now this protein will then produce or it will become a substance, like for example a pigment, wherein itong pigment will influence or will make yung eye color ng isang individual black, brown, or brown. So ganun yung parang pagkakasunod-sunod from the gene up from protein, and then yung paglabas nitong trait or yung paano nabuo itong trait. So, with that, yung unang part or yung unang step in protein synthesis, it is called transcription. So, yung transcription, it is the process by which yung DNA is copied. So, copied, transcribed to mRNA or yung messenger RNA natin, which carries the information needed for protein synthesis. So, yung ginagawa sa transcription, yung DNA, yung gene, gagawa yan, ikakopy, itra-transcribe para makabuo itong mRNA na to. So, ito yung parang important na dapat mabuo dito sa transcription, itong mRNA. So, itong mRNA na to, ito kasi yung nagdadala ng parang code or genetic code para makapagbuo ng protein. So itong mRNA na to, ito yung parang sinusunod, yung code na nandun sa mRNA, yun yung sinusunod ngayon para makapag-produce ng mga protein, mga specific proteins. So yun. So doon yung transcription. Transcription, it takes place in 3 steps. So, tatlo yan na steps sa transcription. Initiation, elongation, and then termination. So, yun yung tatlong steps sa transcription. So, first, let's discuss doon sa initiation. So, in initiation, in the beginning of the transcription ito, it occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. So, tignan natin dito sa diagram. So, itong black part, itong parang ladder na to, imaginein natin ito yung DNA. Then, a part of it, ito yung gene. So, imagine na lang natin na ito yung gene. Now, sa initiation part, yun yung start ng transcription, yung RNA polymerase. Yung RNA polymerase, it ends with A, so you know that it is an enzyme. So itong RNA polymerase na to, itong pink na to, pupunta ngayon yan, magbabind ngayon yan doon sa part ng gene which is called the gene promoter. So yun yung initiation, first part ng initiation. Laging tatandaan lang na yung gagawin is yung RNA polymerase, yung enzyme na RNA polymerase, magbabind ngayon doon sa gene promoter. Yung gene promoter, nandun yun sa, matatagpuan yun doon sa specific part doon sa gene. So, doon siya magbabind. Tapos, after binding, this signals the DNA to unwind so the enzymes can read the bases in one of the DNA strands. The enzyme is ready to make a strand of mRNA with a complementary sequence of bases. So, itong after nag-bind yung enzyme na DNA polymerase doon sa gene promoter, yung gagawin ngayon yan, matutrigger ngayon yung parang yung DNA to unwind. So, di ba, dati ito mga to nagko-connect sila. So, unwind meaning parang nag-loosen up nag-loosen yung DNA chain, yung DNA, kaya parang may nabuong gap, parang may nabuong dalawang strand na. So ganyan, isang strand then isang strand. So yun, nag-unwind yung DNA kasi nga nag-bind yung DNA polymerase doon sa gene promoter. So ginagawa ito para yung DNA polymerase mabasa niya yung bases. Mabasa niya yung ano yung nandun sa let's just say itong mga block na parang stick na to, ito yung babasahin ng DNA polymerase. Itong mga ito, iyon yung mga bases, yung mga glycine, arginine, mga ganun. So, yun yung ginagawa. Kaya nag-unwind para mabasa niya itong mga to, itong mga black stick na tinatawag, which is called the bases of the DNA strand, itong isang strand na to. So, after that, the enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. So, after niya mabasa itong mga bases na to, ready na itong DNA polymerase para makapagbuo ng mRNA. mRNA na it complements yung mga sequence of bases na nabasa niya. So, laging tatandaan yung mRNA, yun yung parang important na dapat na mabuo sa protein synthesis para makapagbuo talaga nitong protein. So, yun. After binding to the gene promoter, nag-unwind yung DNA which lets may parang dalawang strand na nabuo, and then this allows itong DNA polymerase to read para mabasa niya yung information, para mabasa niya yung bases. After niya mabasa, after niya malaman yung bases, then ready na siya para makapagbuo ng mRNA na nakukomplement niya yung mga bases na nabasa niya dito sa DNA strand. So, yun yung initiation. Then, yung palang, di ba, nag-unwind yung DNA. So, may dalawang strand na. So, let's first discuss yung tinatawag na template strand. Kasi ito yung babasahin, no? mRNA, ito yung i-read niya. So, yung template strand, anti-sense strand yon. Then, you also have yung non-template strand, which is yung sense strand. So, ito yung importante, yung anti-sense strand na tinatawag. So, itong anti-sense is the non-coding DNA strand of a gene. So, a cell uses anti-sense DNA strand as a template for producing messenger RNA or yung mRNA that directs the synthesis of protein. So, yun yung tawag dito sa pinapasa ng DNA polymerase. Antisense strand, which is, yun yung ginagamit niya para makapagbuo nitong messenger RNA natin. So, with that, tuloy tayo dun sa explanation. Okay, but before that pala, i-explain ko lang para hindi tayo malito doon sa explanation pa, yung continuation ng explanation. So, sa DNA, doon sa structure ng DNA, it is read in a specific direction just like letters and words in the English language, which are read from left to right. So, each end of the DNA molecule has a number. One end is referred to as the 5', dito sa part na to, and the other end is referred to as 3', dito sa part na to. So, the 5' and the 3' designations refer to the number of carbon atoms in a deoxyribose sugar molecule to which phosphate groups bond. So, yung ano lang yan, yung tatandaan lang dito is yung structure ng DNA is that one part, yung sa left part ng or one of the ends of the DNA, meron ka nitong 5' na tinatawag. So, itong 5' na to, Ito yung, kasi may 5 carbon dyan that is bonded or nagbabind dito sa phosphate group. So, merong 5 na carbon. So, yan. Then, doon sa end naman sa other end ng DNA, meron kang 3' na tinatawag which contains 3 carbon. That is also, na nakaband doon sa phosphate group. So, yun lang yung yung tatandaan muna dito sa DNA structure in relation to protein synthesis. One part of the DNA, meron yan 5' na end, and then doon the other side, doon the other end ng DNA, yun yung 3' na tinatawag. So, yun. So, with that, let's continue. So, after yung initiation. So, summarize muna natin yung initiation. Yung initiation, yung DNA polymerase nag-bind doon sa gene promoter. Now, because yung nag-bind na yung DNA polymerase doon sa gene promoter, nag-unwind ngayon yung DNA. So, with that, essentially nakapag-produce ka ng strand. So, dalawang strand. Now, yung DNA polymerase, since na-unwind na yung DNA, it allows it to read yung isang strand, to read the information, to read the bases. So, kung nabasa na niya yung information or yung bases na nandun sa DNA, sa gene, it is now ready for, to make yung mRNA natin. So, with that, dumako naman tayo doon sa second part ng transcription, which is yung elongation. Now, the RNA polymerase after initiation will move downstream along the genes. So, as the RNA polymerase moves, it will synthesize the mRNA. So, yan. So, itong DNA polymerase, di ba, nabasa na niya. Nag-initiate na siya. Mag-elongate yan. It will move downstream na tinatawag. Yung pag-move ng DNA is from the 3' end ng DNA into the 5' end ng DNA. So, ganun yung movement niya. So, essentially, parang as the diagram implies, from left to right. From 3', magmumove siya hanggang sa 5'. So, habang nagmumove siya, yung RNA polymerase, itong enzyme na pink na to, binabasa na niya. Binabasa na niya itong mga bases. Itong mga stick na to, it is called the bases. So, after that, the DNA polymerase reads the antinescent strand, ito, yung in-explain ko kanina, from 3' to 5'. And then, generating the mRNA from 5' to 3' in which it attaches RNA nucleotide as it moves. So, yun. Di ba, nagmo-move siya from 3' to 5'. As it moves, it will attach itong mRNA na, itong yellow na to or gold na to, ito na yung mRNA natin. So, ina-attach na niya yung corresponding or complementary bases na binabasa niya. So, itong base, let's just say, itong base na to is nabasa niya as yung A base. So, yung ibubuo niya dito sa part na to will be the T base. So, ganun rin dito. If yung nabasa niya dito is C base, then yung ibubuo niya dito is yung G base. So, ano nga ba itong mga bases na to? So, I will explain later. Basta, yung gagawin dapat nitong DNA polymerase is that it will make an mRNA from 5' to 3' that will complement the bases that comes from the antinescence strand. So, yun yung gagawin niya. So, that's the step. And then, after that, ito pala yung sinasabi ko kanina. Yung after that, nakakabuo na siya ng mRNA. So, as the yung DNA polymerase moves from the 3' to the 5', nakakabuo na siya nitong mRNA natin. So ano nga ba yung RNA na tinatawag? So yung RNA is a ribonucleic acid that helps in the synthesis of proteins in our body. So yun yung RNA. So this nucleic acid is responsible for the production of new cells in the human body. It is usually obtained from the DNA molecule. As I've explained kanina, di ba? Yung DNA molecule through the DNA polymerase, from initiation to elongation, nakabuo siya nitong RNA, specifically yung mRNA. Now, ito yung sinasabi ko kanina ng mga bases in which dapat kinocomplement yung mRNA dapat yung bases na nandun sa DNA strand. Dapat pareho sila or complement with each other. So ano nga ba yung mga bases na ito? So sa DNA, ito yung cytosine, guanine, adenine, and thymine. So yung cytosine, dapat yung mape-pair up lang sa kanya is yung guanine. So hindi pwede na yung cytosine, it will pair up with adenine or it will pair up with thymine. So ganoon rin sa guanine. Dapat yung ma-pair up lang sa kanya is yung cytosine at hindi pwedeng ma-pair up yung guanine doon sa adenine or doon sa thymine. So yun yung sinasabi kong complementary bases. So as you can see, GC, CG, GC, CG. So yun yung mga complementary bases, cytosine and guanine. Now, itong adenine and thymine naman, sila naman yung dapat magkasama. So, yung adenine, it will, dapat ito yung kasama niya, itong thymine, at hindi pwede itong guanine or itong cytosine. So, as you can see here, TAAT. So, yan yung DNA. Sa DNA yan, yung base pair, dapat. Yung mga bases. So, laging tatandaan yan. Cytosine with guanine, and then adenine with thymine. So, with that, dito naman sa sa RNA, yung pagkakaiba lang is yung thymine mapapalitan ngayon ng uracil. Pero ganun pa rin yung base pair dapat. Yung cytosine and guanine sila yung magkasama, and then adenine and uracil sila yung magkasama sa RNA. So meaning yung as the DNA polymerase moves dito. So just imagine itong itong red dot na ito yung DNA polymerase. So pag nag-move siya dito, nabasa niya na DNA ito ay na G ito. G na base, itong guanine, nabasa niya na guanine, then yung RNA na ibubuo niya, yung isang base niya is ipe-pair up niya sa cytosine. Then as it moves along, nabasa niya na cytosine yung sunod. Then, yung i-pair up niya, yung base na ilalagay niya dapat doon sa RNA, sa mRNA, should be guanine. Kasi sila yung magkasama, sila yung base pair. Then, as it moves along again, ganun rin, nabasa niya na guanine. Then, dapat cytosine yung nandun sa na base na itatapat niya doon sa RNA. And then, it moves along, cytosine again. Then, guanine yung itatapat niya or yung base pair dapat doon sa mRNA. And then, nabasa niya na nabasa niya na adenine. So, nabasa niya na adenine, then yung dapat i-pair up niya is yung uracil natin kasi RNA yung binubuo niya. So, ganon yung complementary bases, ganon yung buo niya ng mRNA doon sa elongation part na tinatawag. Then, after niya mabuo itong yung mRNA, ito na yung mRNA. So, after niya mabuo, termination na yung magaganap. So, yung termination, once the RNA polymerase reaches the end of gene termination, the end of a gene, termination will occur. So, yun. Pag na-reach na niya, wala na siyang pwedeng pag-move on, then termination will occur. So, the enzyme will detach from the gene and the DNA returns to its original state. So, yan, madedetach na lahat. Yung enzyme, hindi na siya magbabind doon sa gene, and then yung mRNA, madedetach na rin, and then babalik sa dati itong gene na to. So, mag-aano ulit ito, agtipon da ulit itong strand. So, or agbind ulit sila. So, madedetach itong DNA polymerase, and then matatanggal rin itong mRNA natin. So, by the end of termination, an mRNA has been produced. So, yan yung transcription na part. So, yung end goal mo dapat, meron ka nitong mRNA. Dapat nakapag-produce ka ng mRNA. So, ulitin ko, yung transcription, yung first part, initiation. Sa initiation, yung DNA polymerase magbabind sa gene promoter. Now, after binding, this will initiate yung DNA to unwind. So, basically, nag-loose yung DNA strands para it will allow the DNA polymerase to move and to read yung strand para mabasa niya yung yung mga bases. So after that, then it is ready to make the mRNA. So it will move from the 3' end of the DNA to the 5' end of the DNA. So as it moves, binabasa niya yung pair and then kino-complement niya. So pag nabasa niya na C yung nandun na pair, dapat i-complement niya ng G. Pag nabasa niya na A yung nandun sa pair, dapat yung ibubuo niya doon sa RNA na base na i-complement niya is U or yung uracil. And then if it is G ulit yung nabasa na, then dapat C. So ganun. Then after niya mabuo, after niya ma-reach yung end, then termination will take place wherein madedetach ngayon yung madedetach yung DNA polymerase and madedetach rin yung mRNA, and yun, the mRNA has been produced. So yun. So after that, translation naman yung magaganap. So yung mRNA that carries the code in the gene, it will leave the nucleus and go into the cytoplasm where it will find a ribosome in which it will be the start of translation. So meaning, yung transcription, it takes place doon sa nucleus, and then pag nakabuo na ng mRNA, pupunta ngayon yan doon sa cytoplasm, maglalakbay sa cytoplasm in which it will find or maghahanap siya nitong ribosome. So, yun. So, sinasabi dito na yung mRNA carries the code in the gene. So, yun. Kasi nga, due to transcription, di ba? Binabasa niya, kinakopy niya. Parang kinopy niya yung code ng gene. Kaya meron ka na nito. So, basically, parang kinopya niya yung isang strand ng DNA ng gene. Kinopya niya. So, nandun na yung code. Nandun na yung code in which yun yung itra-translate ngayon para makapagbuo ng protein. So, yun yung mRNA. So, yung mRNA from the nucleus pupunta sa ribosome wherein translation will take place. So, before we go deeper into translation, let's talk about yung mga terminologies muna bago natin yung translation part. So first na terminology na dapat nating malaman is yung codon. So codon is a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA. So ito yung kinopya. Ito yung kinukopya nung DNA polymerase kanina para tapos nilalagay niya dun sa mRNA. Ito yung codon. Ito yung genetic code na kinokopya. So, yung codon, sequence down ng 3 nucleotides. So, ito yung GCU, ACG, GAG. Ito yung mga nucleotides. So, 3 sequence. So, tatlo yan. So, a codon comprises of 3 nucleotides. So, itong nucleotides na to, itong codon na to, may pinoform yan na parang specific na amino acid which will in turn become a protein. So yung GCO for example na codon natin, it will code for the amino acid alanine. Yung ACG natin, it will code for the amino acid threonine. GAG, it will code for the glutamate. CUU, it will code for the leucine. So yun. So yun yung codon. Basta sequence of tatlong nucleotides that will code for a specific amino acid. Then another terminology is yung anticodon na tinatawag. So yung anticodon naman, it is a sequence rin of 3 nucleotides on the tRNA naman molecule that bond to the complementary sequence on an mRNA molecule. So yan, yun yung anticodon. So a group of 3 bases on a tRNA molecule that is complementary to 3 bases of an mRNA codon. So yun yung anticodon. Parang parang codon rin siya pero matatagpuan or nakikita dun sa tRNA. Tapos, itong anticodon na to, it complements yung nucleotide na nandun sa mRNA. Meaning, if ito yung codon na nandun sa mRNA, ACC, adenine, cytosine, then, yung anticodon, it will complement yung codon doon sa mRNA. So, when we say complement kanina, in-explain ko, when adenine, dapat yung ka-base pair niya, yung pair niya dapat is uracil. Tapos yung C, cytosine, yung C natin, yung ka-pair niya dapat is yung G. Then C, G. So, hindi pwede na yung A is yung pair niya dapat is yung G, or hindi pwede na yung C, yung pair niya dapat is U. So, dapat complements at kino-complement ng anticodon itong codon natin. So, yun yung anticodon. It is found or kakibat nito yung tRNA or nandun sa tRNA. So, ano nga ba yung tRNA? So, yung tRNA, this is the overall role of the tRNA in protein synthesis is to decode specific codon of mRNA. So, dinedecode niya using its anticodon. So, in order to transfer a specific amino acid to the end of a chain in the ribosome. So, yun yung ginagawa. Kailangan talaga nitong transfer tRNA para parang minomove niya yung transfer nga, minomove niya yung amino acid from one place of the ribosome to another end para ma-allow ulit yung other tRNA to bind with the mRNA. So yun yung transfer RNA or tRNA na tinatawag. Meron siya nitong anticodon which codes or dinedecode itong codon dito sa mRNA. So with that, balik tayo doon sa translation part. So yung during translation, the mRNA acts as a code for a specific protein. So ito yung code na sinasabi doon sa mRNA, which is yung sequence nito na tatlo, it is called the codon. So this happens because each set of 3 bases in an mRNA will code for a specific anticodon which will be carried by the tRNA. So yun, ito yung codon, and then yung tRNA naman, it will complement it with the anticodon niya. So, yung A, it will pair up with the U will pair up with the A, the G will pair up with the C, the U pair up with A, and so on and so forth. So, yon. Then, each tRNA is linked to a specific amino acid na. So, di ba yung mRNA, yun yung na-produce sa transcription. Now, with the tRNA, with the help of the tRNA, dinedecode niya ngayon itong codons na nandito sa mRNA. So, after niya ma-decode, yung decode na sinasabi, yung kino-complement niya, yung mga bases, yung nucleotides. So, kino-complement niya. So, nakapagbuo rin siya nitong anticodon. Now, this anticodon, it is linked to a specific amino acid. So, like for example, yung UAC na anticodon, it is linked with the amino acid methionine. Yung ACG, it is linked with cysteine. UUC, it is linked with lysine. So, sinasabi, each tRNA is attached to a specific amino acid. So, yon. So, we also have, nga pala, yung mga, sa codon pala, we also have what we call as a start and stop codon. So, yung start codon, yun yung AUG, and then yung stop codon, to yung mga UAA, UAG, UGA. So, ang inaano lang nito is during translation, palaging nag-i-start sa codon na AUG. So, dito sa AUG na to. So, nag-i-start siya dapat palagi dyan. So, meaning, yung una or yung palaging na produce na amino acid is methionine. Then, yung stop codon niya is yung UAA, UAG, UGA. So, meaning, mag-i-stop lang yung translation process if nakabasa or may nakitang codon na UAA, UAG, or UGA. So, yun yung start and stop codon. Nag-start, then nag-stop. So, yun yung stop and start codon. So, with that, yung translation, sinasabi, di ba, yung mRNA, it will parang transfer or move from the nucleus into the ribosome. So, it will occur in the ribosome. So, sa ribosome, pwede nating i-divide into two parts. Yung small ribosomal subunit and then the large ribosomal subunit. So, the small ribosomal subunit will bind with the mRNA and a initiator tRNA which will bind to the start codon. So, isa-isahin natin. So, yung mRNA natin, itong mRNA natin, magbabind siya ngayon doon sa small ribosomal subunit, yung part ng ribosome. So, isang part ng ribosome ito, yung small ribosomal subunit. So, magbind silang dalawa, yung mRNA at saka itong small ribosomal subunit. Now, we have here what we call as initiator tRNA. So, yung ginagawa ng initiator tRNA, magbabind siya ngayon doon sa start codon natin. So, laging tatandaan, yung start codon natin is yung AUG. So, after magbind sa AUG, it will complement it, yung base pair. So, G, dapat C, then U, dapat A, then A, dapat U. So, itong UAC na to, it is linked to the amino acid methionine. So, meron ka ng methionine. Then, the large ribosomal subunit will join. So, itong large ribosomal subunit, magjo-join siya to complete the translation initiation complex, and then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. So meaning, after mag-join, after mabuo yung small subunit at saka yung large ribosomal subunit, magjo-join ngayon yung next na tRNA. after the start codon. So, di ba, ito yung AUG natin. Ito yung start codon natin. Kung ano yung pangalawang codon, then, yung tRNA na susunod, magbabind siya doon. So, dapat complement rin. So, yung A natin, dapat i-complements yung U, C, G, GC. So, yung ACG, it is linked to the amino acid cysteine. So, meaning, after nag-bind yung initiator tRNA, tapos yung pangalawang tRNA, meron ka nang nabuong methionine and cysteine na amino acid na nag-link yan. Parang nag-bind yung dalawa. So, methionine and cysteine na. And then, this will carry with it amino acid that will bind to the methionine. Yun yung sinasabi ko. And then, the first tRNA will then detaches, leaves the ribosome, and the whole mRNA shifts over, making room for the next tRNA. So, di ba, kanina magkasama ito, UAC at saka AGC. Ay, itong dalawang tRNA na to. Then, after that, madedetach ngayon yung initiator na tRNA. Madedetach yan para itong second na tRNA natin, magmumove siya ngayon. Magmumove siya, and then, after nakapag-move, meron ng space dito. So, meaning, this will allow the next tRNA, which is yung UUC, mayroon siyang base nucleotide na UUC or meron siyang anticodon na UUC, pupunta siya ngayon dyan, magbabind ulit siya doon sa mRNA. So, dapat, inold ko, dapat nag-complement silang dalawa. So, UUC, dapat yung complement niya doon sa mRNA natin, AAG. So, yon, nag-bind siya doon. So, yung anticodon natin na UUC, it is linked with the amino acid lysine. So, with that, meron na tayong methionine, cysteine, tapos lysine. So, ganun yung mangyayari. The process continues all the way down into the mRNA. So, nagmo-move yan. As tRNA enters and exits the ribosome, exits the ribosome, a polypeptide chain will grow and will continue until a stop codon will be reached. So, yun yung process. After may dalawa na nag-bind, di ba? Madedetach yung nauna, and then magmo-move yung pangalawa. It will take magmo-move siya, and then papasok yung susunod na tRNA with its anticodon, and then it will produce yung amino acid until meron ka na nitong long polypeptide chain na tinatawag, which is chain ng mga amino acid. Then, mag-i-stop lang yung process if meron nang nakita or nabasang UAG, or yun, yung stop codon natin, which is UAG. Isa doon is yung UAG. So, pag meron na tong UAG na codon, then mag-i-stop na yung translation. So, meaning, wala nang mabubuo or wala nang magbabind na tRNA. So, tapos na yung translation. So, the polypeptide will then float away into the other cell organelles for folding and modification. So, basically, yung polypeptide na yun yung parang, di ba sinabi ko kanina na yung amino acid, yung protein, it is made up of amino acid. So, yung amino acid natin, these are the building blocks of protein. So, essentially, yung nabuo na during translation ng mga amino acid na nagbabind, na it becomes polypeptide, this will become eventually yung mga protein natin. So, with that, natapos na yung protein synthesis natin. So, ito yung summary niya. So, sa transcription, DNA, to a series of steps. So, kung hindi nyo maintindihan, then please go back or please watch. Marami dyan sa YouTube, maraming nag-e-explain talaga in simple terms kung ano yung transcription. Basta yung transcription with a series of steps, yung DNA, meron niyang mabubuong mRNA during transcription. Now, yung translation, yung mRNA na nabuo doon sa transcription, ma-decode yan. Using yung tRNA with its anticodon, ma-decode yan. And then, makakapagbuo ng mga amino acid. And then, yung mga amino acid, magiging protein. So, yun yung summary nitong protein synthesis na tinatawag. So, with that, that ends my video lecture on the first part nitong chapter na to sa protein synthesis. So, with that, thank you for listening.